I won't go into a lot of details about adaptive sampling after all the talks that we had yesterday. I will only like to say that adaptive sampling is a fast and flexible target sequencing approach that allows to accept or reject molecules in a real time, only based in the initial part of the sequence. So basically, when the strand enters the pore, the first 500 bases will be read. And based on this information, the algorithm will decide if the strand is likely or unlikely to contain the region of interest. And if the strand is unlikely to contain the region of interest, it will be simply rejected. So our objective is to use this approach, adaptive sampling, to decipher the genetical and functional diversity of the NLRome in melon. This NLRome is the collection of NLR genes that are the most important family of resistant genes in plants. And these NLR genes are very diverse, very variable, even between individuals or varieties of the same species. So it is clear that a single reference genome is not enough to characterize all of them, because we have a lot of NLR genes in other varieties that are not included in the reference. So for that, we plan to sequence around 550 varieties. In fact, we will do 144. We will do adaptive sampling, so we will focus only on the NLR genes. And that's why we're talking about pan-NLRome or NLRome. And we are not talking about pan-genome when we do this collection. I like this figure because we can really see what we plan to do during the project. First, we have our selected varieties. We will sequence them using adaptive sampling, focusing on the NLR genes. And with this information, we plan to do a pan-NLRome graph and obtain and do a variant calling, so obtain SNPs and structural variants. And the final objective is to use all this information to perform genetic association studies and like this, investigate the role of its NLR gene in the expression of immunity of the melon plants that will lead in a future to the creation of new varieties with a high degree of resistance. These NLR genes are, as I said before, the most important family of resistant genes in plants. They are intracellular immune receptor proteins, and they confer a broad range of resistance against a lot of pathogens like fungi, bacteria, insects, or virus. They have a very conserved structure, so they are mainly formed of two domains that are a nucleotide binding site and a leucine-rich repeat. And what is the most important to understand the project is that they are usually grouped into clusters, a cluster of genes that include a high level of presence-absent polymorphism. And also these clusters, they include a large amount of repetitive elements. So in our case, we are using the variety ANSO 77 as the reference. This is a Spanish variety. We produced the reference genome of this variety in our team using also nanopore reads. And we characterize very well this variety regarding NLR genes. As we can see here, we predicted 87 NLR genes that are grouped into 15 clusters. And these clusters, we can see in the figure, they are very variable in terms of size of the cluster and also in terms of number of NLR genes. But what is the most important is that all the clusters, they have a size that is around 2% of the whole melon genome, in concretely 1.6%. And also for each cluster, we give a 20 kilobases buffer zone to ensure that we get the highest coverage in our region of interest. So our first experience was just to test if the approach is working. We decided to sequence the 15 clusters of ANSO using itself as the reference. For that, we used a gridiron. We divided the flow cell in two parts. In one, we did adaptive sampling, and in the other, we did standard sequencing just for comparisons. And we performed, in fact, two types of experiment. In one, we provided a bed reference with just the 15 clusters. And in the other, we masked the repetitive elements that are inside these clusters of genes. We did this because we hypothesized that masking the repetitive elements that are contained in our region of interest will avoid having reads that start in repetitive elements that are kind of similar to the ones inside our regions, but are located off target. So once we did this first experience, then we passed to the Promethean to generate a higher volume of data. And we started to multiplex varieties. And in fact, nowadays we are doing 10 varieties in each flow cell. So talking about results, we can see here that we have a good enrichment of our region of interests. Here I saw 4 of them, 4 clusters, but it happens the same with the 15. On the left, we can see two clusters that are shorter, one of the shortest, and on the right, we see one of the longest. But we can see that it works independently of the length of the cluster. We have always a good coverage on our region of interest and a low coverage in the rest of the chromosome. And if we talk about the statistics, we calculated the enrichment using this formula here, where we take into account the coverage in our region of interest, also the coverage in the rest of the chromosome, and we normalize it with a standard sequencing approach. And we can see that the enrichment, it's kind of variable between clusters, but what is the most important is that it's very high for all of them. And if we take only into account the coverage inside the region, we can see that using adaptive sampling, we get an enrichment of 3-4-fold. That sometimes it's even higher. And if we talk about the assembly, here I saw the assembly of the reference of ANSO, and we can see that we get a high complete and continuous assembly that is also very accurate with the recently released Kit14. So we don't get practically any polymorphism, and with the reference. And what is the most important here to see is that in the assembly that we generate, we find all the predicted NLR genes that are also found in the whole genome assembly. And one question can be, what happens when we try to sequence another varieties that are more genetically distant from the reference? So here I see that I saw you that it works too. I saw 4 of the varieties that we already sequenced. And for these 4 varieties, we have the whole genome. It's published or we produced it, but it's good. We have it, so we can compare. And we can see that for all these varieties, we get also a complete and continuous assembly. And also we find all the NLR genes that are also found in the complete assembly, in the whole genome. And even for some varieties, like Bedrante, that we can see here in green, we find in this assembly built using adaptive sampling more NLR genes that are found in the whole genome assembly. So this means probably that adaptive sampling can help to assemble regions that are highly complex, like they are these NLR clusters. And here I saw a first attempt that we did to build a pan-NLRome graph. For that we used here only 7 varieties, and we used the tool Minigraph Cactus. It's only the first attempt that we did, so we still need to work on this. But probably what we plan to do is to use this pan-NLRome graph as a reference for variant calling. So talking about the perspectives, we advanced quite a lot from the submission deadline, so some of them are already completed. But what we planned in a short term is to evaluate the available methods for pan-genome graph construction. Also, we plan to evaluate the capacity to detect structural variants and copy number variation. And in a long term, we plan to do the same using a different species that has a higher number of NLR genes and also more complex clusters like vineyard. So as conclusion, nanopore adaptive sampling is a simple, reliable, efficient, and cost-saving approach. It doesn't require any difficult standard extraction or library preparation. There is even no need of policing with the Kit14. The volume of data that we generate is highly reduced compared to a whole genome assembly, and it's probably powerful to correct errors in the assembly that are found with the whole genome approach. So what can we do with this is that it allows to explore the genetic and functional diversity of the NLRome in melon using a large amount of varieties. So thanks to Nanopore for giving me such an amazing opportunity of being here today. And also thanks to all of you for being here, and to both of my teams and my PhD directors. Thanks a lot.